So this lecture, we're going to go into this new section, and this is basically the special senses, mostly focusing on the eyeball and the ear. So we're going to talk a little bit about sensory receptors and then go into the special senses. I'm not going to cover taste and smell, but I will go over vision, hearing, and equilibrium. Okay? There is a little bit of a glare. Sorry, this is going to be weird. Just going to adjust it just a tiny bit, just so it's not so much of a glare. We have a glare because the light that comes through the windows. Oh, now that's bad. Sorry. Okay, all right, so we're gonna skip ahead. So there's different types of sensory receptors. There's peripheral sensory receptors, and there are two major categories. There's free nerve endings. So remember we talked all about the structure of neurons, how they have cell bodies, dendrites, axons, axon terminals, boutons. There's two major types of sensory receptors in the body, and that's free nerve endings of sensory neurons. Those monitor general sensory information. So what would be considered general sensory information? General senses, not special senses, just general. What? Touch, perfect. Tactile sensations, right? Touch, vibration, pressure, pain, temperature, all that stuff. That's all general sensory information. Complete receptor cells, these are specialized cells or little mini small neurons, and these are actually complete receptors, and sometimes they take on a very, see you later, okay? They take on a very unique role, and they monitor some certain type of stimuli. These are all involved in the special senses. So these are complete receptor cells or miniature neurons. They're not just free nerve endings that go out into the skin, out into the dermis and different parts of the body. But these are involved in things like vision, rods and cones, hearing, hair cells that actually have hairs on them to pick up hearing. Smell, also have little sort of cilia that project out. Taste, and then here are some examples of free nerve endings and corpuscles, Meissner corpuscles that monitor touch and pain and sensation out in the periphery. So these are kind of all the different types of sensory neurons or sensory receptors. Ooh. Okay, so general sensory receptors, these are just nerve endings of sensory neurons, and those monitor touch, pressure, vibration, stretch, pain, temperature, proprioception. Remember what proprioception is? Yeah, sort of spatial. It's location of body parts and stretch of muscles and tendons and where the body limbs and body is in space. So aware of your body sensations and movements, muscle and joint movement out in the body. So this is a diagram right from your textbook that goes over these general sensory receptors. There's types that are unencapsulated or free nerve endings, and then there are types of sensory receptors that are capsulated, sort of encapsulated in little structures that help them monitor their different stimuli. So here's free nerve endings. There's lots of different examples that use these, like thermoreceptors and nociceptors. Do you remember learning about nociceptors ever? Nociceptors are pain receptors. So these are what pick up pain information. Nociceptors, thermoreceptors that pick up heat and cold stimuli, and mechanoreceptors that detect pressure. So things that detect different chemicals that are released when tissue is damaged. Those usually are nociceptors, pain receptors, and they're just free nerve endings that are out in body tissues. There's also some different modified free nerve endings, tactile epithelial cells. These monitor light pressure, so light touch. There's hair follicle receptors. Again, these are free nerve endings that wrap around, remember, the base of the follicle, sort of bulge or the bottom part of the follicle. So hair deflection is what they specifically detect. When the hair deflects or is pulled, they pick that information up. So these are different types of unencapsulated or free sensory receptors. And then there's encapsulated types. So these are all kind of really interesting types out in the dermis, out in the periphery, things like Meissner's corpuscles, Messinian corpuscles that are kind of wrapped around with these myelin sheath cells that wrap around them. So the nerve ending is there and they're encapsulated. And another one is a Ruffini ending or a bulbous corpuscle. These are involved in proprioception, so stretch of the muscle fibers and the tendon. So they monitor stretch, deep pressure, and so those give information on proprioception. So some of these, if we look at a lot of these different structures, this is the skin. Remember, the epidermis is up here. The dermis is down below. Here's the papillary dermis. A lot of these receptors, here we have light touch, little free nerve endings out here, up in the top of the dermis. We have hair follicle receptors, free nerve endings that are wrapping around the follicles. We have lamellar corpuscles, where there's basically lamellar sort of layers wrapping of support cells and myelin to surround the ending, and those monitor deeper pressure. So when the skin gets indented and there's pushing inward, it activates those deep pressure monitors. Let's see what else. Another pressure is the bulbous corpuscle. So a lot of these are monitoring deep pressure or light touch or vibration, and also pain and temperature, that are the free nerve endings that are out sort of at the top, just above the dermis, and in the lower regions right below the epidermis and the top of the dermis. So there's all different types of sensory receptors that are bringing general sensory information to the brain. 3 types of proprioceptors, or proprioceptors that we'll talk about. These are the major 3 types. Muscle spindles. So the muscle spindles are actually unique little nerve endings that measure the length of a muscle fiber. So they monitor muscle stretch. They're embedded in the perimysium between the muscle fascicles. Okay, so now we're kind of layering things we've already learned. Remember the muscle? Muscle itself is grouped into fascicles, or packets of muscle fibers that are enclosed in a connective tissue membrane. So the perimysium is wrapping around those muscle fascicles. Muscle spindles are embedded right around those fascicles. So they actually are strategically positioned to monitor stretch of the muscle. Second type of proprioceptor, and all of these are monitoring sort of stretch and position of the muscles and movements of the body and the limbs, okay? Golgi tendon organs, these are in the muscle-tendon junction. So right as the muscle is giving rise to the tendon, they're right in that location right there. They monitor tension or stretch within the tendons. So they're keeping track of tendon stretch, how much that tendon is actually stretched or taut. And then there's joint kinesthetic receptors. Kinesthetic, like kinesiology, what does that mean? Kinesiology? Study of movement, right? So this is the movement of the joints. It's picking up stimuli or information on the movement within the joint capsules. So these are free nerve endings within the joint capsules. So all those synovial joint capsules that we talked about their anatomy on, these monitor stretch and also pain or nociception in the joint capsules of all the synovial joints in the body, okay? And remember, we talked a little bit about proprioception already. These stretch receptors and pain receptors that are in the joint capsules. We were learning about the joints and talking about like hyperextension of different joints, and you know, in wrestling they use and different types of, like, what is the name? I can't even think of it. Jiu-jitsu. A lot of those, right, where they're relying on stretch of joints and making people tap out.
Judo?
What?
Judo?
Judo's another one. Yes, exactly. All the floor, I would call them like the grasshopper floor martial arts. Like, you don't want it, you get on the floor and they just, and then get your joint in a compromised position. These will get activated and will tell your brain, ooh, that's being stretched too far. You know, it's got to break. You got to dislocate that joint. Don't do that. So these, I feel like, for you guys to know about the human body and muscles and joints and all that, you should know about proprioceptors and sort of how they function, because these are positioned out there to give information about body stretch, joints, movements of the body. So if we look at what some of these look like, down in the tendon, we have the Golgi tendon organ. Here's the sensory fiber. It's got all the little free nerve endings that are embedded out here at the muscle tendon junction, okay? And then we have a muscle spindle, which is this kind of unique setup that's like a spindle-shaped enclosed little structure, okay? And if we flip back and look at these, measure the changing length or contraction, stretch of a muscle. So how much the muscle fibers are actually being stretched. There are different types of sort of sensory nerve endings. There's the efferent, so the outgoing motor component, and then there's the incoming sensory endings, the type they call it 1A fiber that comes in and then wraps around these specialized muscle fibers, intrafusal muscle fibers, and that they're encapsulated in this little spindle apparatus. And however much all of this is stretching, those are stretching too. right? So these are positioned in the muscles, and they send that information to the brain, how much it's actually stretching. So Golgi tendon organ is at the tendon, muscle spindle is embedded in the muscle fibers. And so it's detecting the level of stretch in various muscles throughout the whole body. So we have the muscle spindles and their little intrafusal fibers with the sensory nerve endings wrapped around them to detect stretch, and the tendon organs and joint kinesthetic receptors that are positioned out at the muscle-tendon junctions. So all of these are giving information about muscle stretch, tendon stretch, and also tendon stretch and also pain. So specifically, joint kinesthetic gives you information about, again, pain. And those are in tendons and joint capsules. So they're free nerve endings that are kind of situated around the joint capsules and in the tendons. Gives a lot of information and allows your brain to collect proprioception information. Okay, so those are, you could do a whole class on all those actually. But I just wanted to give a little bit of those, the general, the most important ones, these different sensory receptors and how they operate in the body. Now I want to switch really to the special senses. So we'll cover these as well. We'll go into them in more depth in physiology. So I want you to have an introduction to the anatomy and function of the eye and the ear. So those are the two that I'm going to focus on. So I have a lot of silly little pictures of my daughter Lola. She was always eating and acting silly, and I have a lot of, so photogenic. So the special senses are sight, hearing, or vision, hearing, balance, taste, and smell. And if we actually said these in more scientific terms, it's vision. Do you know what we call hearing? Like scientifically, we would call hearing? It's called audition. Like auditory sense, not like you're auditioning for a talent show, but audition. Balance, what's a more scientific word for balance? Equilibrium. Taste, do you know that one? Gustation, it's gustation, like gustatory sense, it's gustation means taste. Smell, do you know that one? Olfactory receptors, olfaction, yeah. Vision, audition, equilibrium, gustation, olfaction. But you can say them simply as those 5 special senses. So we have 5 special senses. Some people would say we have 6, or they will say touch is a special sense. It's not, it's a general sensory stimuli that comes in the periphery of the body. All of these 5 special senses are confined to the head, and they have special sensory receptors or specialized little neurons, crazy looking little neurons. Lots of them have hairs projecting off the top, or they respond to different types of stimuli. They're all in the head. They're not free sensory nerve endings. They're different. They're specialized, complete receptor cells. They're like all in one. Stimuli comes in, they get activated, they send that information to the brain. So they're neuron-like cells, and they transfer that sensory information to the other neurons in afferent pathways that ultimately go to the brain. So whole pathway that ultimately goes to the brain. And then your brain integrates and makes sense of that information, okay? All right, so we'll start with the eyeball, the first of these 5 special senses, and one that we're gonna spend a bit of time on because we're gonna dissect this in lab as well. We're gonna do an eyeball dissection. This is the visual organ, the eye. This is my son's little eyeball when he was just a little tiny little toddler. 70% of the body's sensory receptors are in the eyes. 70%. What does that say? If you're thinking about the neurons of the brain and the sensory structures that are involved in these different modalities, what does that tell you when you hear something like that? 70% of all the sensory receptors that are in the body are in the eyeballs. If we add them up, all the different, numbers of sensory receptors or neurons. What does that tell you? There's a lot of them, okay? Yeah. Right.
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 Adapt, but
right.
70% of your.
Right. So if you're blind, that's a major loss of sensory receptors that are working, right, or not working in that case. Many of them are not. It says that our eyes are very important, right? Your eyes and vision, taking in visual cues and stimuli, is very important for us as animals, as humans, right? 40% of the cortex, remember we talked about the outer brain, the cerebral cortex, it has a lot of the higher functioning brain roles. 40% of that cerebral cortex is involved in processing visual information. So not just taking it and going, oh, that's a rainbow, but we take in that information, we make sense of it, we decide, have we seen it before, have we not, is it a person, place, or a thing, is it familiar, is it not, let's make a choice about what to do with that, does it make us happy or sad or trigger something else? Your brain is constantly processing visual stimuli. So we are very visual creatures, okay? Only 1/6 of the eye surface is actually visible. So that's why eyeballs look so strange. If you ever see eyeballs, and like we're gonna dissect them, and it's a big round, sphere. We're only seeing about 1/6 of the surface, and just a little bit of the cornea, and mostly iris. Most of it is back inside of the orbit of the skull, right? You learned all the bones that surround and make up the orbit. So we have a few different accessory structures. Whenever we learn about an organ system, if you see accessory structure, what does that mean? Accessory. If you're an accessory to murder, I don't know, that's random. If you're an accessory to something, what does that mean? You help, or you're like assisting, okay? These are all like the assisting structures. They do not allow you to see, but they help the eyeballs and organ function. Eyebrows, coarse hairs on the superciliary arches. Remember, we got these little superciliary arches. Orbicularis, oculi muscle goes around. Those actually, what do you think the eyebrows do? We could add eyelashes in there too, that come off the palpebrae, your upper eyelid. What do eyebrows and eyelashes do?
They don't want stuff in the eye.
What'd you say, Emily?
They like help protect the eye.
Yeah, they help protect the eye. Seems weird, but I mean, it's like you got little rooftops, right? You got little rooftops that are hairy, help if things are coming down, help to sort of catch dust, particulate matter things. Eyelashes as well. Debris, aerosol stuff is all over the place, floating around, dust, particles, pollen. How many of you guys get really bad allergies? Some of you, I do. Gets in my eyes, gets up my nose, and then just, I'm triggered. That's it. I will have allergies for the next several hours. So the eyelids we call palpebrae. These meet at the medial and lateral angles called canthi, the little pink tissue, sort of right in the inner aspect, you have that. And then this reddish, that it's called the lacrimal caruncle, that little elevation at the medial campus. So you have the little canthi, the medial and lateral canthi are the edges where the two palpebrae come together. The lacrimal caruncle is that pinkish elevation in the medial aspect. Then you have tarsal plates, little connective tissue that's within the eyelids. So eyelids are actually called palpebrae. Okay, so these are all structures to protect the eyes, guard them from a lot of dust and particulate matter getting in them. If they get damaged, you can't see, right? You could actually scratch the cornea, damage the eyeball itself, get infections in the eye. There are infections that affect the eyeball. We also have conjunctiva. Speaking of infections, conjunctiva is actually, so if we look at all this, we've got the little tarsal plates, little connective tissue within the eyelids. You've got eyelash hairs coming off of the palpebrae, the upper and lower palpebrae. And then you have the eyebrow and the superciliary arch that's right here. And then you have you have the levator palpebrae superioris muscle that goes right in and attaches to the tarsal plate. What do you think that allows you to do? This levator palpebrae. To like open and blink your eyelids, right? So when I open or blink my eyelids, I can control it with the muscles that are embedded to the tarsal plates of the palpebrae. There's a conjunctiva, like I mentioned. It's a thin membrane, transparent mucous membrane that's made of epithelial cells that's very thin. It goes up under the palpebrae, coats the entire exposed surface of the eyeball, and then up underneath the lower palpebrae. This conjunctiva is actually a thin little epithelial layer that goes right across the surface. Have you guys ever heard of conjunctivitis? No? Have you heard of pink eye? Same thing, right? Conjunctivitis is infection or inflammation of the conjunctiva. It gets red and kind of gets lots of kind of hemorrhaging and pinkness, and sometimes there's excess fluid, and it makes a lot of eye boogers, I guess you could call them, mucus production, like we call them eye mocos. Basically, it's just when you get pink eye, your eyes get really pink and irritated, inflamed and itchy and watery, and then they start to gunk up in the little medial canthi, little eye crust. That is what happens in pink eye. It comes from streptococcal infection, getting in the eyeball and infecting and inhabiting the conjunctiva. I know this quite well. I've had it in my life. Kids often have it. Pink eye spreads real easy because they itch their eyes and they rub their eyes and then they touch something else, or they touch you and then you get it and it's all over the place. my daughter when she was just about 2 weeks old, newborn infant. Her eyes, one of her eyes got real pink and leaky and gooky and eye mucus, and it was stuck together and have to like pry it open in the morning, and then she had a fever. I think I told you guys about this. Maybe I did not. They thought she might have spinal meningitis at the age of 2 weeks because her fever was going really high, 103, 103.5, 103.9. So I took her immediately to our pediatrician, and they said, oh my gosh, she's got a fever. It might be pink eye, but if it's spinal meningitis, we got to do a spinal tap. We got to get some of that cerebral spinal fluid out of her spinal column and do tests, and we had to be hospitalized. She had to be in the hospital for like 3 days. It was awful. Doing a spinal tap on a tiny little 2-week-old infant, when I was like, it's pink eye, I'm pretty sure it's pink eye. The reason you can't take a risk when you get an extreme high fever like that in an infant is spinal meningitis. You guys are probably, excuse me, sorry about that. You guys are probably aware pink eye actually, I mean meningitis can lead to blindness and deafness and all these different things, right? It affects the cerebral spinal fluid and the meninges. So it can be a really serious thing. But conjunctivitis often happens, and it's affecting the conjunctiva of the eyeball. We also have other sensory structures like the lacrimal gland. Okay, lacrimal gland is up on the lateral aspect, embedded in the above the palpebrae. It actually makes saline solution, or a salty water fluid solution. So tears, it keeps the eyeball lubricated. So there's the whole lacrimal apparatus, which is the gland, and then the little ducts, the punctum, there's a little hole that's right there, and then a canaliculus, a little canal, and then basically it dumps into this lacrimal sac, which ultimately dumps into your nasal cavity. So it dumps into the nose area, this whole nasal cavity, and then ultimately can drip out the nares, right, the nostrils. So this whole thing is called the lacrimal apparatus. It allows the fluid to be produced around the eyeball and then ultimately drain into the nasal cavity. You have that little punctum right there, that little hole, that tiny little hole. Have you guys ever noticed that little tiny hole in your medial aspect? Basically, when you cry and you get that emotional response and trigger of crying, you make a lot of the fluid, the lacrimal fluid, and it drains out of your eyeballs. But also, I don't know if you guys have ever noticed this, you get a really runny nose when you cry, and you get, especially if you're really crying, right? And it comes out, ends up, you get runny nose because it's being produced from that lacrimal gland and then draining out your face and out your nose. I've done that many times. So that's why I said it. That's why when you cry, you get a leaky nose or a runny nose. All right, you've got 6 muscles that control movement of the eye. Again, very, very important. A lot of cranial nerves that we talked about control those extrinsic 6 muscles that control eye movement. They originate in walls of the orbit. They insert on the outer surface of the eyeball. So they attach right to the cornea, the white part of the eyeball. And those muscles are the lateral rectus and medial rectus, superior rectus, inferior rectus, superior oblique, and inferior oblique. And you have to know them all. And we'll be able to see them in lab and look at them as well from a lot of these different figures and then in real life in lab. So we've got the lateral rectus, medial rectus is in the medial aspect, superior oblique, okay, superior oblique, and then inferior oblique. Obliques kind of wrap around going in the oblique direction, right? So they go and back, and then and back. So inferior oblique, superior oblique, and then lateral and medial rectus. If we look over here at the frontal view of the eyeball, superior rectus, inferior rectus, lateral rectus, medial rectus, inferior oblique, okay? And then superior oblique that goes back like this, and then all the way back. These all help to control the rotation and movement of the eyeball. So moving it up, down, abducting, adducting, and so forth. This goes over exactly what muscle it is, what the action it is, and then what cranial nerve controls it, or synapses on that muscle to be able to allow that movement to happen. We talked a lot about these cranial nerves already. Now this is saying exactly which extrinsic eye muscles they control. All right. So those, and we'll get to look at those in lab. So you'll be able to see and dissect away the fat and sort of see those different muscles and how they attach to the eyeball. The anatomy of the actual eyeball, it's got a whole bunch of different components. It's got, all of these need to protect and support the photoreceptors and gather and focus the light on the back of the eye, the retina. So you can make precise images. The retina's in the back of the eye, right here. The lens is right here, and the front cornea, the white part, is right here. So you've got an anterior pole, posterior pole, external walls, and there's 3 layers or tunics that make up those walls of the eyeball. and then there's an internal cavity that has fluids in it. Now the fluids in the eyeball we call humors. So eyeball fluids, all of these are called, not like your humor is your funny, but these humors are actually the fluids in the eye. And whenever you see in the eyeball tunics, you know what a tunic is? Like a sweater, like a Mr. Rogers like sweater vest, a tunic. This means a layer. So when you see tunic in anatomy, that means layer, and the eyeball means layer, okay? Yes? Okay. So if we look at these, the most external layer of the eyeball, if we look at the different three tunics, the most external one has two regions of connective tissue. It has a sclera and it has a cornea. So the sclera is most of the eyeball. It's about 5/6 of this particular tunic. It's the white opaque part. So all the sclera of the eyeball is white. The cornea is the anterior 1/6 of the fibrous tunic, and that's right in the front, and it's clear. So it's one with the sclera in the layer, but it's clear. Sclera is opaque and white, okay? Cornea has to be clear because we need light to be able to come into the eyeball, right? And then we have a limbus, the junction between the sclera and the cornea, the white part versus the clear part. And then we have a sinus, a venous sinus that allows the humor to actually drain. So there's a little hole and a little sinus that picks up and drains the fluid that's in there. It allows it to drain and circulate. So here's a big giant view of the eyeball, and this shows sort of those different layers, parts of this outer tunic that we're talking about. Cornea, okay, sclera, all the way around. Yes? So that's sort of where it is. So cornea is right out front. Conjunctiva would be the thin layer that goes around this entire thing like this. The scleral sinus, the venous sinus, is this little hole right here that allows the fluid to drain. So the fluid drain gets resorbed, more fluid gets made, and it kind of recycles. So it has this whole waterway draining system. And then we go to the fluids in the eye. In the back behind the lens, so here's the posterior pole of the eyeball, here's the anterior pole of the eyeball. There's this humor in the back here, it's called the vitreous humor. This is really thick and it's kind of like gelatin. And you'll see when we cut our eyeballs, we're gonna cut our eyeballs in half and we're gonna kerplunk that out. It actually looks like Jell-O. It's thick, sort of viscous gelatinous fluid, and you can plop it right out. We're gonna look at that in lab. Okay, so the vascular layer, this is the next innermost layer. This is the middle tunic or layer of the eyeball. We have the choroid, the ciliary body, and the iris. You know where the iris is, right? Everybody knows where the iris is. What's the iris? Right, it's the colored part of your eye, right? And there's genes that color code exactly what combinations of colors that iris is, okay? Choroid is the vascular, darkly pigmented membrane, okay? And it forms the posterior, the back 5/6 of this vascular layer. It has a brown color to it, and it's brown color because it has melanocytes. So this is back in the back part of the eye. The choroid is this kind of brownish melanocyte layer of this vascular tunic. Really, it prevents scattering of light within the eye. So it's brown, it has pigment, and prevents light from scattering all over the place, concentrating those light photons or rays to the back of the eyeball. Yeah?
What's it called? My friend has beautiful eyes and she has one blue, one green. What is it
Ooh.
called?
Yeah, so that is, it's kind of a mosaic. It's like a mosaic of the expression of those genes are different in different tissues. And it's rare, but it happens sometimes. Different genes are expressed in different tissues asymmetrically. It happens in dogs and cats and animals and humans sometimes. It's really neat. There's some people I think that I've seen people with that, like one blue and one brown. It's really, really interesting. So it's a genetic sort of mosaic expressed differently in different tissues. All right, prevent scattering, so the choroid is really important. The ciliary body is also really important. This is this thickened ring of tissue that encircles back behind the lens. The lens looks like a cough drop, okay? So when you're dissecting the eyeball, I always get students saying, what is this? What is this? This is the cornea? It's not the cornea. It looks like a little throat lozenge, like a cough drop. It's the lens. It actually looks just like a nice little kind of rounded camera lens. And this is back behind the lens. It's made of a ciliary muscle and ciliary processes, and also a zonule, which I like to call the suspensory ligament, because the suspensory ligament sort of describes exactly what it is. The suspensory ligaments are attached around the entire outer periphery of the lens. So they help like little cables, little ligaments, to hold the lens in place. And they actually allow the changing shape of the lens, we'll talk about later. So these suspend the lens and allow it to change shape when focusing on near versus far objects. Your lens has to bulge up when looking at something close and flatten out, skinny out when you're looking at something far. The ciliary body with the processes and the suspensory ligament allow your lens to stay attached and to bulge or flatten when it needs to do that for viewing different objects. This actually is called accommodation. So accommodation is something that allows your lens to accommodate to seeing or having visual acuity of something close or far away. And as you get older, that actually goes down because you lose the elasticity of the epithelium of the lens. It gets harder, it gets less stretchy and less elastic. So for me, even as I age, this refers to how close you can see something in clear view when it's close to your face. So we'll do these tests in physiology, but basically it allows you to focus on near versus far objects. And as